Module B, Introduction to Gatekeeping Strategies. In this module, I will provide a general introduction to an important class of testing strategies that have been developed to address multiplicity issues in clinical trials with complex multivariate objectives. Efficient solutions for multiplicity problems of this kind can be obtained using so-called gatekeeping strategies, where the multiple analyses are grouped into families and tested sequentially. We're going to begin with general regulatory considerations in section B1 and a brief summary of key points from the draft guidelines recently published by the US Food and Drug Administration and the European Medicines Agency. I will introduce a simple classification of gatekeeping strategies. These includes, for example, main types of gatekeeping strategies known as serial, parallel, and general gatekeeping strategies. In section B, we will go over other relevant regulatory considerations in confirmatory trials with predefined secondary analysis. For example, issues arising in the evaluation of treatment effects on secondary endpoints, power calculations, analysis of secondary endpoints across several trials within a single phase 3 development program. Section B1, Gatekeeping Strategies. In very general terms, gatekeeping strategies are employed in advanced multiplicity problems that we encounter in confirmatory trials with several families of clinical objectives. This slide displays a typical hierarchy of multiple objectives in a confirmatory trial, and most of the time when we deal with confirmatory phase 3 trials, we discuss the process of grouping those objectives into the three categories that are displayed on the slide using orange boxes. Those objectives, specifically primary, secondary, and exploratory objectives, are typically arranged from the most important ones at the top to the least important ones at the bottom. And on the next slide, I'm going to go a little bit deeper into the definition and role of primary, secondary, and exploratory objectives in confirmatory trials. How do we define the primary, secondary, and exploratory objectives in general confirmatory trials? Primary objectives are the objectives that make or break a clinical trial, as we sometimes say, which means that the primary objectives are directly related to the trial's outcome. And for this reason, they are presented in product labels using the so-called inferential statements. This means that whenever we see a summary of findings, results from the primary analysis in the product label, it is typically accompanied by a p-value, which signifies that the observed effect is statistically significant, and in addition to that, a confidence interval may be attached to the summary of the results. And similarly, when we look at secondary objectives, also known sometimes as key secondary objectives, those provide key supportive evidence of treatment effectiveness, and they are typically also presented in product labels using inferential statements, which again means that we will typically see a summary that includes p-values and or confidence intervals. By contrast, when we move on to summaries of exploratory objectives that used to be known as other secondary objectives, we know that they play a general supportive role, and they're typically summarized, presented in product labels using descriptive statements. Instead of using p-values or confidence intervals, the product label would focus on descriptive statistics or a plot. This includes, for example, survival curves that may be used in product labels that summarize results from phase 3 oncology trials. It will be appropriate now to give a couple of quotes from the FDA guidance on multiple endpoints in clinical trials. Section 3A of this guidance document provides a good discussion of the role of primary endpoints and secondary endpoints in a confirmatory setting, and these two quotes that you see on the slide support the general statements that we went over on slides 6 and 7. The only quick point I would like to make here is that this FDA guidance focuses on multiplicity induced by the analysis of multiple endpoints. And this is why it always refers in those quotes and throughout the documents to endpoints. But we need to recognize that the same principles would of course apply to other types of primary and secondary objectives. For example, to settings where the primary objective may be formulated in terms of treatment effectiveness in the overall trial population, and the key secondary objective may be related to the treatment effect within a pre-specified subset of this population, which may be based on a certain biomarker. Coming back to the quotes given on the slide, the FDA guidance gives the following definition of the primary and secondary endpoints. Endpoints essential to establishing effectiveness for approval are called primary endpoints. Secondary endpoints may be used to support the primary endpoint or endpoints and/or demonstrate additional effects. This guidance document emphasizes that positive results on the secondary endpoints can be interpreted only if there is first a demonstration of a treatment effect on the primary endpoint family, which means that, as I'm going to explain in a couple of minutes, that the primary endpoint family serves as a gatekeeper for the other endpoints, the other objectives, based, for example, on the secondary endpoints. It will be instructive to return to the diagram from slide 5. We said that inferential statements based on p-values and/or confidence intervals can be formulated based on the primary and secondary analysis, and it would be natural to apply a multiplicity adjustment only to the top two boxes in this diagram, to the box that includes the primary objectives and to the box that includes the secondary objectives. This overarching multiplicity adjustment is represented graphically on the slide using a dashed box. And when the overall type 1 error rate or alpha is controlled with respect to the pre-specified primary and secondary objectives, this can be accomplished using appropriately defined gatekeeping strategies. This alpha control can be used when the overall type 1 error rate or alpha is controlled with respect to the pre-specified primary and secondary endpoints, and this can be accomplished using appropriately defined gatekeeping strategies. We will talk about this later in this module and in other modules. So this alpha control can be used to justify the inclusion of the secondary findings in the product label. And when we look at the bottom box that corresponds to the exploratory objectives, there is clearly no need to provide alpha control. Remember that we said that exploratory analysis play a supportive role, and the results of exploratory analysis are presented in product labels using descriptive statements, not inferential statements, and for this and no p-values can be presented, and for this reason there is really no need to even consider alpha control. This last point is emphasized in the FDA guidance on multiple endpoints in clinical trials. Quoting from section 2E of this guidance document, presenting p-values from descriptive analysis, that is, from analysis that were not pre-specified and for which appropriate multiplicity adjustments were not applied, is inappropriate because doing so would imply a statistically rigorous conclusion and convey a level of certainty about the effects that is not supported by that trial. For this reason, we draw a line between the three boxes shown on the diagram on slide 9, and alpha control would be required only for the top two boxes. No alpha control is applied to the exploratory objectives, and as a result, presenting p-values from those analyses in the product label would not be appropriate. Gatekeeping procedures. With this slide, we're beginning to talk about commonly used solutions to complex multiplicity problems in confirmatory trials with several families of primary and secondary clinical objectives. This is accomplished using testing strategies based on gatekeeping procedures. So let us begin with the simple question. How do we define gatekeeping procedures? The definition is given at the top of this slide. In very simple terms, gatekeeping procedures are simply examples of multiple testing procedures that are designed for this particular class of multiplicity problems that employ multiple families of null hypotheses. An important property of gatekeeping strategies is that they are based on the closed testing principle and they preserve the type 1 error rate, known typically in this context as global family-wise error rate control. I would like to emphasize the word global because in this case our goal is not to reach for local control within each family of null hypotheses, but instead of that, provide that overall overarching global control over all multiple families that include hypotheses corresponding to the primary as well as secondary objectives in the clinical trial. The reason it is important, as we emphasized a few slides back, is because with global family-wise error rate control, we can safely provide important information on secondary objectives for prescribing physicians, patients, and other parties without having to worry about compromising alpha control. Another important property of gatekeeping procedures, I should say of effective gatekeeping procedures, is that they respect the hierarchical structure of multiple families and they help maximize power by counting that structure. The way they're set up is that they focus on providing more power for more important objectives. And then whenever the primary objectives have been met, we can figure out how much alpha can be transferred to the secondary objective, which may potentially lead to lower power for those secondary objectives, but this will be consistent with the predefined hierarchical structure. As we've looked at the examples of hierarchical diagrams, we always recognize the fact that the primary endpoints or primary objectives are almost infinitely more important than the secondary objectives, and therefore it will be in this trial sponsor's best interest to first try to maximize power within the box of primary objectives, and then whatever resources are left would be applied to the box of secondary objectives. This is a simple diagram that captures the main idea behind any gatekeeping strategy. Gatekeeping strategies, as I said on slide 11, support efficient solutions for multiplicity problems where multiple analyses are grouped into families, and these families of analyses or objectives or null hypotheses of no effect are tested sequentially. For this reason, every family except for the last one in this predefined sequence of families serves as a gatekeeper for the next family in the sequence. So family I serves as a gatekeeper for family I plus 1. And we will discuss the specific conditions under which the gatekeeper, which is family I, can be passed. That includes parallel, serial, or general gatekeeping conditions, and this will be done later in this module. There are numerous examples of successful applications of gatekeeping strategies in confirmatory clinical trials. One of my favorite examples is the use of powerful Hommel and Hogberg-based gatekeeping procedures in the lurasidone phase 3 development program in schizophrenia. In fact, example 3 that was introduced in Module A is actually based on one of the confirmatory trials from this program. I will present general methods that are employed to set up gatekeeping procedures similar to those that were developed for this lurasidone development program in Module D. And I would like to emphasize that the importance of using gatekeeping procedures in the individual trials within the lurasidone development program was emphasized in the clinical trial publications. You see the references on the slide. And there are multiple other examples. Gatekeeping procedures have been successfully developed in several, I would say numerous phase 3 trials or programs. This includes, for example, new treatments for the treatment of insomnia or type 2 diabetes programs. There are also several industry-wide guidelines that stress that it is critical to prospectively define strategies for addressing multiplicity induced by the analysis of several outcome measures as well as other sources of multiplicity. To give one example, the recommendations released by the Initiative on Methods, Measurement, and Pain Assessment in Clinical Trials state that to decrease the probability of a type 1 error in pain clinical trials, the use of gatekeeping procedures and other methods that correct for multiple analyses is recommended when a single primary endpoint does not adequately reflect the overall benefits of treatment. Classification of gatekeepers. We will begin with two useful classification schemes that help us better understand the structure of various gatekeeping strategies in clinical trials. The first classification scheme deals with the classification of gatekeepers, or families of hypotheses that serve as building blocks in more complex multiplicity settings. What we see on the slide is a list of commonly used gatekeepers. They are known as serial gatekeepers, parallel gatekeepers, and general gatekeepers. And in modules C and D, I will provide multiple examples of phase 3 clinical trials that give us examples of families with those gatekeeping conditions. Serial gatekeeping conditions, parallel gatekeeping conditions, and finally general gatekeeping conditions. The list of references here is far from being comprehensive. It does not even get close. A lot of work has been done in this general area over the past 15 years, and I wanted to include those references mostly to help you get more information about those popular classes of gatekeepers. In addition to this, there's another classification schemes that approaches this problem from a slightly different angle. Here we're going to talk about the difference between sequential testing strategy with retesting, and finally I will say a few words about simultaneous testing strategies with retesting. This slide defines probably the most popular approach of connecting gatekeepers. Here we assume that family 1 is a gatekeeper. This could be, as we said on slide 17, a serial gatekeeper, a parallel gatekeeper, or a general gatekeeper. And provided the appropriate conditions are met, we can pass this family 1, this gatekeeper, to proceed to carrying out tests that are included in family 2. This will be an example of families that are tested sequentially, and the error rate, alpha, will be transferred along the sequence. We're going to apply all of the alpha that's available to us to test the hypotheses included in family 1, and then, once again, provided the appropriate conditions are met, we will find out how much alpha can be carried over to family 2. The simple sequential testing strategy can be enhanced by including a retesting loop, which is illustrated in this diagram. As you see on this diagram, we have the same two families, labeled family 1 and family 2, and there are two arrows that connect those families. The first one that connects family 1 with family 2 is exactly the same type of alpha propagation strategy that we discussed on slide 18. But in addition to the simple, straightforward sequential testing strategy, we can also enable the retesting loop. And this retesting loop will be utilized if certain hypotheses were not rejected in family 1 the first time when we carried out the corresponding tests. For example, if family 1 serves as a parallel gatekeeper, we can pass this gatekeeper if only one hypothesis out of potentially many hypotheses is rejected, and therefore the sponsor may be interested, after examining the hypothesis included in family 2, the sponsor may be interested in returning to family 1 and retesting those hypotheses that were not rejected the first time around. As a result, this testing strategy enables a clinical trial sponsor to transfer the error rate, or alpha, along the sequence and then back to family 1. The last example of testing strategies that could be envisioned in phase 3 clinical trials would be a simultaneous testing strategy with retesting. In this case, as indicated in the diagram, families 1 and 2 would be tested simultaneously, and if certain hypotheses are not rejected within each one of the families, there is an option to transfer the alpha back and forth between families 1 and 2. That's why it's known as a simultaneous testing strategy. At this point, I am not aware of any phase 3 clinical trials that have utilized this testing strategy. However, my colleagues and I have written a paper on a family of gatekeeping procedures known as superchain procedures that may be used to implement this simultaneous testing strategy in a real-life phase 3 trial. Serial gatekeepers. We are now going to take a closer look at the commonly used types of gatekeepers that we encounter in phase 3 clinical trials. As I said a few minutes ago, those popular classes of gatekeepers include serial gatekeepers, parallel gatekeepers, and general gatekeepers. And in this case, we're going to assume that we're dealing with a simple two-family multiplicity problem in which family 1 serves as a serial gatekeeper for family 2. And the serial gatekeeping condition is formulated at the bottom of the slide. We say that family 1 is a serial gatekeeper for family 2 if all null hypotheses must be rejected within this gatekeeper to proceed to family 2. It turns out that the serial gatekeeping condition is quite straightforward in the sense that it is very easy to build gatekeeping procedures that support this serial gatekeeping strategy. In this case, we're going to assume a slightly more general setting where there are m families of hypotheses, and we will assume that the family Fi serves as a serial gatekeeper for the next family in the sequence, which is denoted by Fi plus 1. Once again, all hypotheses must be rejected within Fi to proceed to the next family in the sequence. Gatekeeping procedures that satisfy this serial gatekeeping condition can be constructed in a very straightforward fashion. All we need to do is to carry out all tests within a serial gatekeeper at the full alpha level. If we assume that all of the families here serve as serial gatekeepers for the next family, then this approach would be applied to families 1 through m minus 1, and any procedure can be used in family Fm because this is the last family in the sequence. With this approach, it's quite easy to show that the resulting serial gatekeeping procedure controls the global family-wise error rate at the alpha level. As a quick illustration, we're going to return to example 1 based on the phase 3 trial in patients with prostate cancer that was introduced in module A. And just to refresh our memory, in this example, the primary hypothesis was based on overall survival, and it was accompanied by 3 predefined secondary endpoints that are denoted on the slide by S1, S2, and S3. For each one of those endpoints, we can define the hypothesis of no effect, and they're denoted by H1 through H4. If we assume now that there are 4 families in this clinical trial, and each family includes a single hypothesis, then this results in a very straightforward serial gatekeeping strategy. This strategy is visualized on the slide, and in this case, to be able to proceed to the next family in the sequence, it is sufficient to simply reject a single hypothesis within each individual family. And all it boils down to is a simple sequential testing approach. This is what we refer to as the fixed sequence procedure in part 1 of this online training course. Under this approach, each hypothesis is tested at the full alpha level, provided of course that all preceding hypotheses are rejected. Parallel gatekeepers. I explained in the preceding section that if we impose a straightforward serial gatekeeping condition to the multifamily problem of interest, it is quite straightforward to build serial gatekeeping procedures. All we need to do is to carry out each individual test at the full alpha level. Now, within this section, we're going to take a look at a more flexible testing strategy, and due to this flexibility, we will find out quite soon that we'll need to spend a bit more time on setting up valid multiplicity adjustments that are known as parallel gatekeeping procedures. The general structure of a simple two-family problem is presented on the slide. We assume again that we're dealing with two families of null hypotheses, and if family 1 is assumed to be a parallel gatekeeper for family 2, this implies the following condition. One or more null hypotheses must be rejected within this parallel gatekeeper, or family 1, to proceed to family 2. As we saw on slide 27, the parallel gatekeeping condition enables more flexible testing strategies, because to be able to pass a gatekeeper, we no longer require that all null hypotheses must be rejected within that gatekeeper. It is sufficient to reject a single null hypothesis. I would like to emphasize that from the sponsor's perspective, it may be desirable to reject as many hypotheses in that primary family as possible, but formally, to be able to satisfy the appropriate regulatory requirements, it is sufficient to reject just a single null. Example 2, based on the type 2 diabetes trial, serves as a very good example of a setting where a parallel gatekeeping strategy would be quite meaningful. In this case, as I explained in Module A, the 3 dose or regimen placebo comparisons considered in this clinical trial are grouped into families. The family of primary comparisons include the comparisons of the fixed doses of saxagliptin versus placebo. The corresponding null hypotheses are denoted by H1 and H2, and also in addition to this, another secondary family is defined, and this secondary family includes a single hypothesis. This hypothesis, H3, is formulated in terms of comparing the up titration strategy, this regimen that includes potential up titration of saxagliptin 2.5 milligrams to the 5 milligram dose, and this regimen is compared to placebo. It is very helpful to visualize the parallel testing strategy used in example 2 as using a diagram shown on this slide. This diagram shows us that within the primary family there are two hypotheses, and once again within the secondary family there is just one hypothesis, H3, and since the primary family serves as a parallel gatekeeper for the secondary family, it is sufficient to reject only one of the two hypotheses, H1 or H2, included within the primary family, and to represent graphically this testing strategy, I connected the hypothesis H1 to H3 using an arrow, and also there's an arrow that connects the hypothesis H2 to the hypothesis H3. So if we follow those decision-making paths, we can see here that with a single rejection in the primary family, we can claim that we have met the primary objective of the trial, and we can proceed to testing the secondary objectives that are included in family 2. General gatekeepers. Example 3, based on the phase 3 clinical trial in patients with schizophrenia that we introduced in Module A, serves as an example of a clinical trial that relies on an even more complicated type of gatekeeping conditions. Let's begin first of all with defining the hypothesis of interest. If you remember from Module a two-dimensional multiplicity problem arises in this example, in this clinical trial example, and this is due to the fact that there are two clinical endpoints pre-specified, the primary endpoint and the secondary endpoint. And in addition to this, the second source of multiplicity is defined by the fact that there are two doses of the experimental drug compared to placebo. This results in four null hypotheses of no treatment effect, and those are grouped into two families: the family of primary endpoint tests and the family of secondary endpoint tests. Those families are defined on the slide. The best way to visualize the two-dimensional multiplicity problem in this clinical trial example is to construct a matrix-like structure that we see on slide 32. This 2 by 2 matrix has two rows, and those two rows correspond to the two families that are defined by the primary and secondary endpoints, and the two columns in this matrix correspond to the dose placebo comparisons: low dose versus placebo and high dose versus placebo. I've used the dashed boxes to define the two families, and those two families are arranged vertically so that we begin testing with the hypotheses in family 1, and if an appropriate clinically relevant condition is satisfied within that family, we can step down and carry out the secondary tests included in family 2. Now the question that we need to ask here would be, what is the condition that needs to be satisfied within family 1 before we can proceed to family 2? And a closely related question is, what are the most meaningful logical relationships among the hypotheses in the primary and secondary family? To answer this question, it will be useful to think about clinically meaningful logical restrictions that are imposed on the individual hypotheses in similar two-dimensional multiplicity problems. There are several papers that discussed this topic, and here I would like to include a quote and a specific recommendation from a paper that was published in 2009. It is emphasized in this paper that whenever we have to deal with similar two-dimensional multiplicity problems, it is most relevant to consider the following class of logical relationships. It is stated in this paper that a dose placebo test on the key secondary endpoint is relevant only if a significant treatment effect was established for this dose on the primary endpoint. Using this information, let's now come back to the diagram and see what would be the most meaningful way to connect the boxes that correspond to the null hypotheses of interest. The diagram that visualizes the clinically relevant logical restrictions that we discussed just a few seconds ago is presented on slide 34. According to the recommendation that we just went over, it is most meaningful to connect the hypothesis H1 and H3 and separately the hypotheses H2 and H4 as shown on the slide using the black arrows. Those logical restrictions translate into the following general conditions that are shown at the bottom of the slide. According to the clinically meaningful logical restrictions, the hypothesis H3 can be tested only if H1 is rejected, and likewise H4 can be tested only if H2 is rejected. It will be instructive to compare and contrast the logical relationships that we defined on slide 34 with logical relationships that could be induced by parallel gatekeeping conditions. For example, if we try to consider the diagonal, so to speak, logical restrictions or logical connections that, for example, try to connect the hypothesis H1 and H4 and also potentially connect the hypothesis H2 and H3 as shown in this diagram, we immediately realize that those logical relationships are not clinically meaningful in the context of this particular clinical trial. This immediately tells us that family 1 cannot be thought of as a parallel gatekeeper for family 2. That's because the diagonal connections are no longer clinically relevant. This will be an example of a general set of gatekeeping conditions, and for this reason, family 1 will be termed here as a general gatekeeper for family 2. We're going to discuss in much more detail the process of setting up appropriate and valid gatekeeping procedures that are consistent with those general gatekeeping conditions in Module D. Section B2, Other Regulatory Considerations. Now that we have set the stage for the discussion of complex multiplicity problems arising in confirmatory clinical trials, which includes multiplicity problems with gatekeeper and different types of testing strategies, it would be helpful to spend some time on the discussion of some of the more practical issues that arise in the analysis of secondary objectives in clinical trials. And here I'm going to touch upon the topics listed on the slide that are known as pseudo-specificity. I'm going to explain what it is and what role this concept plays in the analysis of secondary objectives. We're going to talk briefly about sample size calculations in clinical trials with secondary analysis. And at the end of the section, we're going to talk about pooled analysis of secondary endpoints. Let us begin with the topic of pseudo-specificity. Pseudo-specificity is an interesting concept that arises in clinical trials with hierarchically arranged multiple objectives, which includes primary objectives and secondary objectives. And in very simple terms, pseudo-specificity reflects the Food and Drug Administration's restrictions on the use of secondary objectives in product labels. Key secondary objectives, for example secondary endpoints, are typically expected to provide additional information on the treatment's efficacy compared to the primary objective. Secondary objectives should not be clinically related to the primary objective. And if a secondary objective is indeed closely related to the primary objective, this secondary objective or secondary endpoint may be referred to as pseudo-specific. The ideas related to pseudo-specificity are intuitively appealing, and based on my experience, pseudo-specificity may take center stage in key discussions around the formulation of effectiveness claims for novel treatments. But there's currently much uncertainty around the specifics. There are really no documents that shed any light on the exact definition of pseudo-specific secondary endpoints, and pseudo-specificity, for example, is not discussed in the FDA guidance on multiple endpoints. I think it will be helpful to illustrate potential impact of pseudo-specificity in the regulatory discussions, and I would like to give two examples. The first example is based on a phase 3 study in patients with a major depressive disorder. In this case, the primary endpoint was based on an instrument which is commonly used in major depressive disorder trials. It is the MATTRS total score, which stands for the Montgomery-Åsberg Depression Rating Scale. The key secondary endpoints included a fairly large family of potentially supportive endpoints. Here on this slide, I would like to focus on the first two of those secondary endpoints. The first one is based on a certain functional impairment score, and the second secondary endpoint was essentially a simple transformation of the primary endpoint. It was known as the MATTRS-based remission status, and patients was classified as being in remission if their MATTRS total score was below 6. You can see here that in this case one can easily argue that the secondary endpoint is directly related to the primary endpoint, and this is where it would be relevant to discuss the role of pseudo-specificity. In this particular case, when this discussion took place at the Food and Drug Administration, the regulatory reviewers argued that the secondary endpoint needs to be removed from the list of secondary endpoints due to pseudospecificity concerns. Essentially, the information based on this secondary endpoint, MADRS remission status, does not provide much new clinical information on top of what can be derived from the analysis of the primary endpoint. The clinical trial example presented on slide 40 appears to be quite straightforward. This is the case where concepts related to pseudospecificity can help guide the process of selecting the most relevant, the most regulatorily relevant secondary endpoints. However, if we try to apply similar ideas to other phase 3 clinical trials, those, for example, that are not necessarily reviewed by the FDA Division of Psychiatry Products, that's where we may discover that concepts related to pseudospecificity are not always directly applicable to the selection of secondary endpoints. Here is a quick example based on a phase 3 development program in patients who experienced episodic migraine. The trials that were included in this development program are known as Evolve 1 and Evolve 2 trials. The primary endpoint, which is defined on the slide, was based on the mean change in the number of monthly migraine headache days, and there was a number of secondary endpoints pre-specified. Here I would like to call your attention to the first two secondary endpoints that were defined as the proportion of participants with a reduction from baseline of over 50% and over 75% in monthly migraine headache days. In this case, one could easily argue that those two particular secondary endpoints are very closely related to the primary endpoint, and they may not provide much additional clinical information compared to the primary endpoint. However, the regulatory reviewers in this case were quite open to accepting those key secondary endpoints, and no references to pseudospecificity were made in the context of this particular development program. I would like to reiterate that the FDA's draft guidance on multiple endpoints does not mention pseudospecific endpoints, but you can find quotes or statements that allude to situations where a secondary endpoint could be closely related to the primary endpoint. For example, you will find this quote in section 3A. It is stated in the FDA guidance that another example is a secondary endpoint of the percentage of patients whose symptoms are very improved, when the primary endpoint is the percentage of patients with any amount of improvement for the same symptoms. Adjustment for multiplicity is necessary to demonstrate these additional effects. This statement approaches the situation from a purely statistical perspective. It essentially states that in certain cases it could be appropriate to consider secondary endpoints that are derived using simple mathematical transformations from the primary endpoint. And if this actually happens, of course, we should not forget about multiplicity that may be induced by the analysis of those supportive endpoints. And of course, the sponsor needs to pre-specify a valid multiplicity adjustment to be able to include those secondary endpoints in the product label. Another potentially relevant statement from section 3D from the FDA's guidance on multiple endpoints. This section states that an example of the second type of component analysis might be found in trials of antipsychotic drugs in which positive and negative symptoms are domains collected in the positive and negative syndrome scale PANSS and often analyzed separately in addition to the overall scale. Interpretation of analyses of any subscale domain, however, is dependent on that subscale domain having been previously evaluated and determined to be valid as a standalone clinical measure. Here we see an indication that selecting meaningful secondary endpoints may be guided by certain clinical principles. Specifically, we may see in this quote that a secondary endpoint may not be valid as a standalone clinical measure. Perhaps it's not that informative by itself and does not add much information in terms of new clinical information to the primary analysis, which is based on the composite primary endpoint. And in this case, this composite primary endpoint is the PANSS total score. Sample size calculations. In most clinical trials, the sample size is based on achieving adequate power for the primary endpoint. However, it is not always the case, and in this section we will explore the following question. Should key secondary endpoints play any role in power or sample size calculations? To answer this general question, it would be useful to take a look at the FDA guidance on multiple endpoints, and it's interesting that this guidance provides a direct answer to the question. It emphasizes that if success on the secondary endpoints is important, the secondary endpoints should be considered when determining study design, for example, sample size. The FREEDOM trial is a great case in point, and it will help us understand the reasons why a trial sponsor may be interested in shifting focus from the primary endpoint to one of the supportive secondary endpoints when running a sample size calculation. This trial was conducted to evaluate a novel treatment, denosumab, for the prevention of fractures in women with osteoporosis. There were 3 endpoints in this trial, a primary endpoint and 2 secondary endpoints that provided supportive evidence of effectiveness. The primary endpoint and secondary endpoints are defined on the slide. The primary endpoint was the risk of developing new vertebral fractures, and it was followed by non-vertebral fractures, which is endpoint S1. And I would like to point out that this endpoint included hip fractures. And finally, the secondary endpoint, labeled endpoint S2, was based on the assessment of the risk of developing new hip fractures. Those 3 endpoints were tested hierarchically in the trial, and naturally the probability of success on the last secondary endpoint, S2, was the lowest because the first 2 endpoints in the sequence served as gatekeepers. But the trial sponsor knew that hip fractures were very rare compared to vertebral fractures and non-vertebral fractures, and to account for this important feature of endpoint S2, a larger sample size was required to have adequate power for this important secondary endpoint. A summary of power calculations in the FREEDOM trial is presented on the slide. The sample size, which was almost 7,900 women enrolled in this trial, was driven not by the primary endpoint. The primary test was clearly overpowered. It was powered at around 99%. The sample size in this trial was selected to ensure a high power on the hip fracture endpoint, which was S2. The resulting level of power for the secondary endpoint was close to 90%. So this would be a good example of a phase 3 trial where the trial is overpowered on the primary endpoint, and the goal here is to guarantee adequate power for a very important secondary variable. And I would like to say a few words about possible interpretation of secondary endpoints that end up playing a central role in sample size calculations. The fact that key secondary endpoints may play a central role in power calculations has led to a number of discussions about the nature, if you will, of endpoint labels. Specifically, is there truly a difference between the primary endpoint and the key secondary endpoint in a trial if the sample size calculation is driven by this key secondary endpoint? Is it true that the line between primary and key secondary endpoints has become blurry? Well, when we think about those questions, even if a phase 3 trial is powered based on a secondary endpoint, there is still an important even fundamental difference between the primary and secondary endpoints. The difference is that the primary endpoint serves as a gatekeeper. This endpoint could make or break a clinical trial, and the secondary endpoint is a supportive endpoint. That's why it's called a secondary endpoint. So I would not say that the line between primary and key secondary endpoints is indeed getting blurry. Coming back quickly to the topic of power and sample size calculations in clinical trials with complex objectives, I would like to remind you that, of course, closed form expressions for power and sample size calculations are not available in this context. We cannot apply an analytical approach, and we are forced essentially to consider simulation-based alternatives. This topic, simulation-based power and sample size calculations, will be discussed in Module G. Pooled analysis of secondary endpoints. The last topic we're going to consider in this module is related to the analysis of secondary endpoints in development programs that include multiple phase 3 clinical trials, which is how most development programs are typically set up. So if we step back and think about the principles for the analysis of primary endpoints in clinical programs with several trials, we know that a strict consistency criterion is applied. In fact, it's mandated in regulatory guidance documents because the beneficial effect must be demonstrated in each trial within a development program. Now, the question we're going to explore in this subsection is the following. When we now try to think about possible approaches to analyzing secondary endpoints in a development program with several phase 3 trials, what are the available options? Option number one will be to analyze secondary endpoints as if this was a primary endpoint, which means that we're going to apply the same consistency criterion. A statistically significant difference would have to be demonstrated on each given secondary endpoint within each trial across the development program. An alternative approach relies on a pooled analysis across trials within a program, and this is a topic that was discussed in a recent publication. When we think about consequences of applying the options defined on slide 52, we realize that option 1 is actually fairly conservative. Option 2 helps address potential limitations of option 1, and advantages of the pooled analysis of secondary endpoints across several trials within a development program include improved power and lower likelihood of observing inconsistent results. Let's begin with the first point. Why should we worry about improved power for secondary endpoints? Even though we talked about the possibility of performing sample size calculations for secondary endpoints just a few minutes ago, in reality, this option is limited to a very small number of trials. I would say that in 95% of phase 3 trials, sample size calculations still rely on the primary endpoint. And it's quite common to see secondary endpoints, secondary analysis being underpowered. From this perspective, if there is an option to help improve power of secondary analysis, I think it will be fair to consider this option. Secondly, if we consider applying a strict consistency criterion to the analysis of secondary endpoints, we realize that we are going to run a risk of observing inconsistent results across the trials, especially if the number of trials within a development program is fairly large. We can address this problem, and we can increase the likelihood of observing positive results based on a secondary endpoint by applying option 2, which relies on a pooled analysis of secondary endpoints across the trials. To help us better understand the pros and cons of those two options, I will quickly introduce a case study based on a phase 3 trial in patients with cystic fibrosis. This new treatment with the brand name Orkambi was actually a combination drug that was approved for the treatment of cystic fibrosis, in a certain subset of the general patient population. This would be an example of a targeted therapy that was effective in patients with a certain mutation in the gene that encodes the cystic fibrosis transmembrane conductance regulatory protein. This is what CFTR stands for. There were two trials included in this phase 3 development program in cystic fibrosis. They were called the Traffic and Transport trial. Within each trial, the main source of multiplicity was the comparison of two treatment regimens for this combination drug therapy that will be labeled regimen 1 and regimen 2, and they were compared to placebo. The second source of multiplicity was the analysis of the primary endpoint, which is defined on the slide, and also multiple key secondary endpoints that are listed on slide 56. We're not going to spend too much time on those secondary endpoints. We're simply going to recognize the fact that clinically relevant secondary endpoints were predefined in both of those two trials, in the traffic and in the transport trial. A very important feature of the clinical study report for both trials that can be downloaded from the FDA's website was the fact that the program's sponsor decided to present the analyses of the primary and secondary endpoints not only within each of the individual trials, but they also presented a very useful analysis based on pooling the data from the two trials. And the table that you see on slide 57 summarizes the results of those pooled analyses for each of the 5 secondary endpoints. You can see here that when we take a closer look at the performance of regimen 1, we see that significant results were observed for all secondary endpoints on regimen 1. I highlighted the statistically significant p-values. And when we switch to regimen 2, we see that significant results were observed for the first two endpoints, S1 and S2. That's a very useful set of results. Unfortunately, when we look at the final set of results from the traffic and transport trials, we realize that the product label presents the key secondary results only within each trial, and those results are summarized in this table. We can see here that unlike the table shown on slide 57, this table indicates that only one of the endpoints was significant on regimen 1 instead of 5 as on slide 57, and when it comes to regimen 2, the results between options 1 and 2 happen to be significant. Here, just like on slide 57, when the pooled analysis was employed, both endpoint S1 and S2 turned out to be significant. We're pretty much done with module B, and I would like to touch upon another important topic directly related to the main theme of this section, related to how we handle secondary endpoints in confirmatory trials. Ordering of secondary endpoints is a critical consideration in most phase 3 trials because secondary endpoints are most commonly tested sequentially using the fixed sequence test. I give you an example based on a prostate cancer trial with 3 secondary endpoints that were examined sequentially. This was done earlier in this module. And with this fixed sequence testing approach, we can carry out the hypothesis tests along the sequence only as long as the tests are significant. The first time we see a non-significant test, this will be the end of the game. The remaining endpoints will not be tested. The corresponding hypotheses of no effect will be accepted automatically. End of story. The EMA guidelines share some considerations related to this topic. The very first sentence of this slide is worth rereading several times. It is stated in this quote that the ranking of endpoints in a hierarchy can be a source of controversy. It is truly the case. I could not have said it better. And then the following statement is made. In principle, the planning and assessment of a clinical trial should prioritize those endpoints of greatest interest from a clinical perspective, but it has become a common practice to rank endpoints based on the likelihood that the individual hypothesis can be rejected. The reality, I would say, is more complicated. I completely agree that it would be ideal to arrange the endpoints from the most clinically relevant in the context of this particular indication to those that are least relevant. But I've seen so many phase 3 trials where the secondary endpoints were grouped into two families: a family of endpoints of greater clinical importance and a family of less important endpoints. It was obvious that the more important group of endpoints ought to be considered first. It would define the family of more important endpoints, but it was not clear how to best arrange the endpoints within each family. Should the endpoints within each family be tested sequentially, or should those tests that rely on a family-driven testing sequence be applied? For example, step-down tests such as the Holm test. Those would be very good questions. So what can we do in real-life trials to address this issue? I would like to quickly share my experiences. I've helped develop optimization strategies aimed at identifying the most reasonable way to arrange secondary endpoints based on clinically relevant criteria. The idea here is to incorporate information on clinical relevance and then enumerate all meaningful testing options. For example, sequential testing versus step-down testing, perhaps some other testing strategies, and then we can select an optimal testing strategy for the secondary endpoints, provided of course that some information on the anticipated effect sizes and pairwise correlations is available. We don't have time to explore this topic in this online training course, but I think it's a very promising direction. I would like to encourage you to consider optimization related approaches when it comes to finding the best way to arrange secondary endpoints in phase 3 clinical trials. Here I will just say that we reviewed practical approaches to clinical trial optimization with applications to optimal selection of multiplicity adjustments, including optimal selection of ordering of secondary endpoints, in a book which is entitled Clinical Trial Optimization Using R.